Yep, so that's a very interesting talk. So we've seen throughout these talks how important antifibrinolytic drugs are, and we saw with Professor Murphy's talk, despite the fact that some of the previous trials of Aprotinin have been heavily criticised, once data is out there in the literature, it's very difficult, I think, sometimes for us to know exactly where it sits. And of course, the regulators have their own problems as well. So we're delighted to welcome Dr. Royston, who's going to come and talk to us about where Aprotinin sits now and what the regulators' current views on its use are. Thank you.
Well, thank you very much indeed. Thanks for the invitation and to the Society for letting me to come to one of your meetings. The interesting thing about this is that this is probably one of the few drugs that's ever been reintroduced into the market. And what I want to do over the next 25 minutes or so is to talk through the processes that went on in the various regulators and why this the license has been given back to Aprotinin and what we will be able to use it for clinically according to the regulators in the future. Basically, I have a few conflicts of interest. I am still a consultant at Bayer until my 5-year term runs out. I am, in fact, the chairman of the Data Safety Monitoring Committee looking at fibrinogen in aortic surgery, but I won't tell you what the answer is just yet. And I'm also a consultant for Nordic, who've taken over the license for selling Aprotinin worldwide. Bayer have basically given that up. Okay, let's start right at the beginning. Why did the regulators come along and say that there was a better benefit-risk ratio for Aprotinin than the other drugs, and therefore it had to be back on the market to be used in clinical practice? The first thing comes from what we call an antifibrinolytic. And basically, if you look at the fibrinolytic system, you have got the fibrinolytic protein is plasmin, and plasmin will act at a certain site on the fibrinogen molecule to split the molecule. What we don't want to do is to have free plasmin. So we have a natural serine protease inhibitor called alpha-2 plasmin inhibitor, which binds to free plasmin with a time of about 100th of a second. It's one of the fastest enzyme inhibitor complexes known in nature. What it doesn't do, it doesn't stop bound plasmin. The lysine analogues, what they do is they fit into the plasmin molecule or plasminogen molecule and stop them binding. So that basically means you get free plasminogen. Right. This is a simple experiment. You get a bunny rabbit, you make a hole in its ear, it bleeds, you give the animal TPA, so that's hyperfibrinolytic bleeding, it bleeds more. You then give the natural serine protease inhibitor, alpha-2 plasmin inhibitor, it stops the bleeding. Okay? But at the same time, you take the rabbit's blood, you allow it to clot, you add the same dose of TPA to that, and it takes about 2 hours, the whole clot will lyse. You put in the same dose of alpha-2 plasmin inhibitor and you get about 85% clot lysis. So your natural serine protease inhibitor, the one that's floating around your body at the moment, will stop you bleeding due to hyperfibrinolysis, but it will not stop you from lysing clot. Your physiological fibrinolysis works. If you do the same experiments using Aprotinin, it does the same thing. It stops you bleeding, it doesn't stop the clot from lysing. It is not an antifibrinolytic, it is an antiplasmin. Okay? What the lysine analog antifibrinolytics do is they do not affect bleeding one iota. What they do is they stop the clot from lysing, and that's all they do. They basically stop the blood that's in your chest coming out the drains, and therefore you think you've got a benefit. Okay. Obviously, with the two differences in mechanisms, you're going to get two different dose-response relationships as well. So basically, if you look at patients having reoperation surgery and the proportion of patients who receive transfusions and the amount of transfusion in hepatic surgery and in major orthopedic surgery, there is a significant dose-response relationship when using Aprotinin compared with placebo or nothing. And the orthopedic surgery one's quite interesting because if in that group they pre-donated blood, and using a very high dose of Aprotinin, you can get from giving an average of 6 units of blood per patient down to about 0. Okay. The lysine analogues have only been studied in one study, was done about 20 years ago, to look at a dose-response relationship. And as you'd anticipate, once you get effective fibrinolysis at a dose of about 2 to 2.5 grams, that is basically the plateau effect. And none of these studies in the early days showed any benefit of transfusions. So essentially, tranexamic acid will work as an antifibrinolytic. It reduces your drains volumes, and because of that, you condition yourself not to give the patient any blood. It's not because of any other reason. So if you now look at the data from a recent Cochrane analysis, and this is what the regulators saw, that the exposure to donor blood is reduced by about 32%. Reoperation, which is its big advantage, is reduced by half. The other drugs, lysine analogues, do not affect reexploration rates because they don't stop that microvascular bleeding. They just stop the drains from coming out, reoperations reduced, and all the rest of it. The other thing that's critically important is aprotinin is the only agent that's been shown to be effective in patients who've taken antiplatelet agents. So there is one study with tranexamic acid and aspirin which showed absolutely no benefit whatsoever in that group of patients. All the studies with aprotinin work with aspirin, and there are 3 randomized studies that looked at aspirin and clopidogrel where again the aprotinin worked. There's no studies in the world literature looking at tranexamic acid and aspirin and clopidogrel treated patients. So the regulators thought, well, we've got a benefit of this drug, and it's got a bigger benefit than giving tranexamic acid, so that's one thing. But is it safe? And here we start on a long and very tedious journey, thanks to the New England Journal of Medicine, who you will eventually guess at the end of this is not my favourite journal anymore, or its editorship. And this started with Dennis Mangano's paper in 2006, which basically came out and said that aprotinin was associated with a bit of a bad problem. And then there were 2 other what's called observational studies that came out, which also said the same thing. And then the randomized placebo, or the randomized control trial, which was known as the BART trial. What I'm going to do is to explain to you why the regulators looked at these studies and decided that they really couldn't show that there was a significant risk of the use of aprotinin. Okay. Now, the problem about observational studies is that they're not randomized placebo-controlled studies. So what you've got to do is you've got to mathematically manipulate the data to make it look as if they're a pseudo-randomized study. That's uses something called propensity scoring. And you can do this in one of 2 ways. Either you can set up a matching process or stratification process where you put patients into silos and then compare those when you know what the treatment is afterwards, or you can use a form of regression analysis. The problem about this secondary thing is if you've got any major imbalances of the background characteristics between the 2 groups, then essentially this is not a useful technique. And basically, observational propensity scores, for those that are interested in Harry Potter, this is really a dark art. Seriously, I've spent lots of time talking to various people who invented this technique, and this technique is really quite interesting. I'll not ask you questions about this afterwards, but this is basically how you define it. And I assure you, I know diddly squat about this, but it strikes me as being, it's got potential for fiddling data. So if you look at the various studies that have come out, Carcuti, I'll mention it right at the end because it's an interesting sort of point, but they use matching. The FDA, the Food and Drug Administration in America, their regulators looked at subclassification. And the other papers that were written in the New England Journal and published in the New England Journal as major articles on observational studies use this multivariable regression technique. Okay. If you first of all look at the Mangano paper, which was the original one that came out, and the FDA looked at this in some detail, he had 47 factors which made his propensity score. And included in that are things that are obviously critically important in whether a patient's going to bleed or do badly after their surgery, like their income, whether they've got a stable relationship with their partner, and such things. The only definition they have of heart failure is if you got given dobutamine. Okay. The FDA didn't really like that, so they went along to the chief of cardiac surgery at Bethesda and says, tell us some of the risk factors for why people do badly after cardiac surgery. And he came up with his colleagues with a number of factors. And when they reanalyzed the Mangano data, they essentially found out that all the things that were published in the New England Journal basically disappeared as significant problems. The only thing that maintained itself was the need for dialysis post-operatively. Okay, and that was really something which nobody could understand until about 2 months after this particular data had been analysed by the regulators. This paper came out looking at the same database, which basically showed the incidence of renal dysfunction and renal failure requiring hemofiltration stroke dialysis in 4 countries. And you can see that on average you get more biochemical evidence of a raised creatinine in 3 countries, but in Germany, where 80% of the Mangano aprotinin patients came from, they actually filter more people than get renal dysfunction. So obviously they're filtering people for things like acidosis or oliguria, not necessarily for any kind of renal impairment. When you put geography into the propensity score, essentially the renal failure signal disappears as well. So the Mangano data, when you analyse it in what is thought to be a more appropriate way, as shown by the regulators, doesn't show any risks of aprotinin at all. What in fact does come out of the data, though, from the Bayer randomized control studies is that there is an increased risk of getting a small rise in creatinine, which is transient and occurs in the 3 to 5 days after the surgery. And that is a given. It does exactly the same thing as radiocontrast or as aminoglycosides, and obviously if you give aminoglycosides and aprotinin together, you get a bigger rise, but it's transient. It goes on for about an average of 9 days instead of an average of 6 days, which normally happens in these other small group of people. So that is a known effect. The regulators have accepted that will go into the product information. And that is the only adverse event that there is, apart from the one that has been around for ages, which is giving aprotinin, again, within 6 months of the first administration, is associated with increased risk of having an anaphylactic reaction. So those are the two safety issues that the regulators agreed on. Okay. Next to that came along was published in February of 2008, and the two papers were published on the same date. So this and the Andy Shaw paper from Duke. This is the Schneeweiser, it's the analysis of this. It's the Premier Perspective I3 database. This is a database which is actually an administrative database, but what it does is it actually works out how much you should charge the patient. So there's a lot of information that's not included in this as well. And this leads to some other problems. But what it does have massive numbers. So they were able to compare about 30,000 patients with aprotinin with about 45,000 patients who received aminocaproic acid, which is the licensed analog used in America. It's not licensed for use anywhere else in the world because it's too dangerous, but it's all right for Americans. It comes up with a number of improbabilities because of this. If you look at the renal outcomes model, you'll see that in their group of patients, their data set when they analysed it came out to show that high blood pressure was highly protective, as was a recent MI, as was if you smoke currently. And these all reach significance statistically. So you can see that this therefore raises some questions compared with more conventional ways of analysing data, such as the Society of Thoracic Surgeons. And in fact, if you look at the mortality signal, which they say was specific for aprotinin, what you had to have to get the lowest mortality is have high blood pressure, smoke cigarettes, have a high income, as obvious, have cancer, recent MI, have a neurological dysfunction, again, a stable relationship with your partner, and be older. And that protects you in their database. So you can see that the regulators looked at this and said, well, actually, because there's so many cofactors missing, we really can't use this data to assess the benefit risk of aprotinin, and therefore they didn't. The Shaw paper, as I say, published in the same journal in February of 2008, is basically from the Duke database, and it doesn't take a genius to work out that you are comparing apples with oranges here. 1,300 aprotinin patients, 6,500 Amicar patients. They've got totally different for emergency valves, the bypass times, and if the transfusion rates as well. So obviously, they couldn't actually match this at all. The regression analyses didn't quite fit. There were lots of factors that were still significantly different between the two groups. What they did publish, though, in the supplement, which is available on the internet, was a matched pair analysis. Okay, so they got about 1,000 patients that they could match, but they were still not matched for transfusion, age, and year of surgery. They couldn't actually match for those factors as well. But overall, if you look at this group of people, they had no effect on mortality at either 1 year or at 30 days. And again, you'll see that the 30-day mortality between 4 and 5% implies that this group of patients were probably on the very sick end of the market by most people's standards. But again, the mortality signal disappears, there is no other safety signal in the data, and therefore the regulators said this paper shouldn't be used or can't show that aprotinin is dangerous. Which brings us along to BART. BART was going to be the thing that threw the situation either way. It was a randomized study. It compared Amicar with tranexamic acid with aprotinin in what they called high-risk patients. Okay? And it was done in 19 centres. It was supposed to have about 3,000 patients altogether. And what happened in 2007, we had a meeting with the FDA about looking at the Schneeweiser paper and looking at the paper from Duke, which again, the FDA said there wasn't a problem, but we'll wait for the BART study. They were told by the BART investigators on September the 12th that there had been 2 interim analyses and there was no obvious problems. On the 19th of October, we were told that the study had been stopped because the Data Safety Monitoring Board had decided that there was a safety signal, and on November the 5th, the marketing approval was withdrawn when the German regulatory authority applied to the EU to have the license withdrawn. Okay. What happened then was that if you work out the logistics of this, basically they'd had 2 interim analyses of about 2,000 patients, and then they had about 400 patients' data which they'd had serious adverse event forms filled in for, but they hadn't got the data books. So they had to wait for that data to come along, to get cleaned up, to put into a locked database, and then analyse the data. So essentially, the last part of the study is a post hoc analysis. They knew what the answer was going to be because they knew what had been published. Okay? And they also had a deadline to do this as well, because on May the 14th, the editor of the New England Journal had to go along to the Senate and explain what a load of idiots the FDA were, because they'd allowed all sorts of drugs which the New England Journal had said were dangerous to be allowed on the good people of America. And they were talking about Vioxx, rosiglitazone, and as they said, and we will show tonight at 5 o'clock when it's the Bart paper published that Aprotinin kills people as well. And obviously, the tort lawyers in America went completely ballistic, and million and billion-dollar lawsuits flooded in because this was it. The New England Journal had said, it's a killer. Right. However, what happened next is still a complete mystery, because what happened was that when the Bart study had been published, the partially redacted data in all the case record forms were sent by the Ottawa Health Institute and Health Canada to Bayer to look at. And I spent my summer of 2008 going through these data books, and it didn't take a genius to work out very quickly what had gone wrong and why the Bart study showed why it showed. Okay, so we went along then to the Health Canada and said, we think you ought to look at some of this because it doesn't make a lot of sense. They set up an expert advisory committee which sat in December 2008, and this is the constitutional body of that. As you can see, they're not people that have got an axe to grind, you would have thought. And basically what they've worked out was there were 137 patients' data which had been excluded from the analysis. And these data had been excluded from the analysis for no reason, because if you look at the way it was supposed to work, they were supposed to analyse the intent to treat group as they were randomised. These patients, 137 of them disappeared, and they published on these data. It just so turns out that there are 11 deaths in these patients' groups, for a mortality rate in that group of 8%. And so basically, that group, when those patients were put back into the study, essentially the mortality signal disappeared. It became non-significant. And that was, quote, not ever satisfactorily explained, why they did take out all these people. Okay. But what they did do was that they showed that also, when they put the patient's data back which had been removed, that they could show that aprotinin was significantly better in this group of patients than was tranexamic acid. Okay? So you got a 30% reduction in bleeding. It was superior to tranexamic acid with a risk reduction of about 18% and about 14% for all blood and blood components. So essentially, the finding that it didn't make any difference was reversed when they put the patient's data back in again. What he also showed as well is there was huge differences between mortality and between centres. And they, Health Canada did a signal analysis to say why is it that we get this actually increase in mortality, because there are more people died in the aprotinin treated group, even though it's not significant statistically. And what they came up with was there were 5 centres where they had absolutely no mortality at all. They had 9 centres which the mortality was roughly equivalent, and 5 centres where there was a huge mortality difference. And what they came up with for the explanation of this is differences in heparin management. And so these people with a high mortality difference use significantly less heparin. Now, that's got the regulators very exercised because that's a big, significantly statistical difference. But I think the biological relevance of giving 47,000 as opposed to 3,000 units of heparin to a patient having cardiac surgery is a little bit lost on me. However, the heparin management has now become one of the focal points of what the regulators want us to look at when we're examining if there are adverse events, because that's the thing that came out of the Bart study. As I say, I don't think there's anything in there. I don't think this is biologically relevant, but that was the signal. Okay, the other thing, and the thing that I basically was responsible for, rather than the statistical part of this, was that there was also a change in reclassification of endpoints. And the panel concluded that this aspect was, again, not satisfactorily explained. So they had the meeting on December the 3rd, and then they had a teleconference with the Bart investigators to say, okay, we've told you what we found, explain why it's turned, and they couldn't explain this. Okay. And basically what they'd done is they looked at the two interim analyses, and as you can see, if you look at the overall endpoint, the primary endpoint to massive bleeding, it's lower in the aprotinin patients than the tranexamic acid patients in the two interim analyses. As I said, there are 434 patients' data books which weren't in this, that were then analysed post-hoc because they knew what the answer was going to be, and this is what happened in that group. All of a sudden, there was a doubling in the rate of problems with aprotinin and a small fall with tranexamic acid. And again, if you looked at the individual components of this is reoperations. The last group doubled the rate of re-exploration, and the tranexamic acid group fell a bit. And basically, the incidence of loss of 1,500 mils in 8 hours virtually disappeared for tranexamic acid and kept the same for aprotinin. And as I say, that was the problem. That essentially, there was a lot of data which seems to have been altered in some way to make the answer come up with what you want it to be. And again, it was never satisfactorily explained, and therefore the regulators decided that you could not use the BART study as it was published in terms of a risk exercise. And therefore they ignored that. So now all of the papers showing an adverse outcome with aprotinin have either been ignored or been shown to have a flaw, and therefore the regulators basically sat down and said, well, we've got all this potential benefit, and actually the risks don't really exist when you look at the data in its entirety, and therefore we'll give the license back. The problem we now have is, what do we use for the patients? Which patients do we use it on? Because Kevin Carcuti, he used a matched pair system he's got a bespoke risk management system that they use in Toronto. And if you look at the BART study, their incidence of mortality is about 2 to 3%, which is quite low. In the moderate risk group in being given aprotinin and tranexamic acid in Toronto, again, there's no difference in the mortality signal, although there is a slight increase in the number of patients given aprotinin who died. But in what they call really high-risk patients who were given aprotinin compared with tranexamic acid, there is a significant reduction in the mortality signal in that group of patients. Why that is, we don't know, because the numbers are relatively small, but essentially that's the group of patients that we would probably want to use aprotinin in, and certainly that's when I first described it in, is reoperation patients and high-risk patients, not for routine coronaries. I don't think it's really got a place in routine coronary surgery, but essentially it's good for that. However, the regulators have given it a license which is not for high-risk patients. So there will in fact be a registry that's going to be developed, and as part of that you're just going to tell the regulators you're using this drug off-label because it's not for first-time coronaries. This is one that came out a little bit later. This is in fact one from Professor Murphy's group, but basically the Bristol data. They had about 6,500 patients. They were able to match two cohorts of patients. If you looked at the all-patient group, then the patients without aprotinin had higher bleeding, which is obviously a given. Re-exploration rates, again, a complete given because that's what it does. But also they got less acute kidney injury, and that's an interesting finding, which again might suggest because they're not being as transfused, they're not getting as much problems. And the high-risk group, again, they showed, as did Carcuti, a significant increase in mortality if you did not receive aprotinin. So essentially what we're up to at the moment is, as I say, there's a registry going to be formed. The Nordic company are going to come along and develop that. That's going to be the way that the regulators are going to assess the problems. And lastly, and not least, the one thing about tranexamic acid is because we don't know what the dose is, that we've now got a whole mass of papers that have come out to say that if you're doing open procedures, i.e. not just coronary artery bypass graft surgery, and you use relatively high doses of tranexamic acid, you increase the risk of having a seizure or some kind of fit in the early post-operative period. As far as we know, that doesn't relate to any long-term neurological deficit, but studies haven't been done yet. But you do, in fact, increase the rate of fitting in the ITU in these patients. The mechanism is really quite well known. The pharmacologist has been using tranexamic acid as a GABA antagonist since it was invented in about 1950, and that's how it works. It is basically a GABA antagonist. So that's the problem with tranexamic acid. So as I say, aprotinin will be on the market hopefully by the end of this year. The Nordic people are here if you want to talk to them about that. That's a commercial decision. They've got to get all sorts of bits of paperwork with the regulators sorted out. But essentially, from our point of view, there is no safety signal apart from transient rise in creatinine, anaphylactic risk if you give it repeatedly, and basically we just got to fill in the registry data to tell them what we're doing and why. Thank you.
Thank you very much, Dr. Royston, for a very educational talk.